Welcome back, everyone. Today we're going to be talking a bit about microscopes. So to start us off, we will be using a light microscope, which how light microscopes work is they shoot a beam of light through two compound lenses, and they use that light, they bend it, they refract it to magnify objects. So we're trying to zoom in. We're trying to look at really small things and make them seem larger. So today we will be using a compound microscope, and some of the main differences between a compound scope and a dissecting scope is that our compound scopes can look a whole heck of a lot closer than a dissecting scope. Compound scopes have a variety of different objective lenses, which we'll talk about what that is in just a second. But they allow us to look at different magnifications of whatever we're looking at. And we can get pretty dang close. We can see individual cells, plant and animal cells, using objective microscopes or using compound microscopes, whereas with a dissecting scope, we would not be able to see that level of detail. So some of the main important pieces of our scopes. We have our ocular lenses and the eyepiece. So this part of our microscope does contain a lens, one of the two lenses, that helps magnify the object that we're looking at. And this is also where we look into the microscope. We also have an arm, that's the back piece here, and a base, which is the bottom here. And these are going to be important when it comes to carrying your microscope and proper procedure there, which we'll talk about. This is our light source down at the bottom right here. So this is what's going to blast that beam of light upward through our microscope, through the stage, which is the stage is where our little slide rests right in here. And we have little metal clips to hold on to those slides. But here, this little turret, okay, of changeable lenses. These are our objective lenses. This is what's going to give you different levels of magnification in your microscope. Okay, so depending on what objective lens you choose. So this is our second lens. If our first lens is this eyepiece, okay, the ocular lens, the objective lens is our second lens. Okay, so that's kind of the main. We also have light control. So there will be a little wheel on the side of your microscope. Now where this is can kind of depend. It could be closer on the back, on the arm. But this is going to determine the brightness of that light that gets shined upward. You'll also see these big knobs on the side. Okay, the big one right here, the big part of this knob, this is the coarse focus. So this is what's going to move your stage up and down and bring your slide into focus. This little part of it, this smaller piece right here, that is the fine focus. So that's going to move your stage just barely to really help you get it perfectly clear, a perfectly clear image with just really fine, delicate movements. So in terms of magnification, we have to take into account our different objective lenses. So on your microscopes this week, you will see 3 to 4 objective lenses. So that's 3 to 4 of these little different lenses we got here. And depending on which lens we're looking at, that will depend what the magnification is. So all of you will have at least these 3. Some of the microscopes we're working with will have an even higher magnification, but we'll talk about that one in class. So our scanning, that's our lowest objective, which means it has the least magnification. So on our scanning objective, you're going to see about 4 times magnification from that lens, which means we're seeing what's on our slide about 4 times bigger than it is in real life. So our low power, that's about 10x, 10 times bigger. And our high power objective, that is 40 times bigger. Now, we have to remember, though, it's not just the objective lens that's doing the magnification, right? We have 2 lenses, the objective lens and the ocular lens. So to get total magnification, we have to do some multiplication. So for our scanning objective, we get 4 times magnification from the objective. We get 10 times magnification from the ocular lens, and you'll see these are always going to be 10 times. Okay, so our total magnification is just 4 times 10, which equals 40. So with our scanning objective, our lowest objective, we are going to see what is on our slide about 40 times bigger than it would be to the naked eye. Okay, same for low, same for high power, same equation, we multiply, and we get 100 times, and we get 400 times magnification. Our highest is going to be even more powerful, but again, we'll talk about that one in class. So we need a few ground rules before we start working with microscopes. Okay, we want to be careful. Now, this is usually where I like to ask the class about how expensive do they think a microscope is. Go ahead, take a minute, take a guess. Okay, but it's pretty dang expensive. We'll have a fun little guessing game in class. But microscopes are expensive, and we need to be careful with them. In fact, the most nerve-wracking time for me is when everybody's carrying their microscopes from the cabinet to their table. So let's get some ground rules. We will always want to respect and take care of our expensive equipment. So first things first, all backpacks out of the way today. Okay, I'll remind you in class, but we want to make sure they're up on the table, out of aisles, so that nobody's tripping over any backpacks. You will find your microscope in the cabinets, and you're going to see it's stored with the cord wrapped around it. The scanning objective will be in place, which I'll show to you guys. And the stage is going to be all the way down. So that's how we find our microscopes in the cabinet. That is how they will want to be returned. Make sure you're returning them exactly as you found them and properly storing them. You'll also notice that your microscope is numbered, and it is numbered to its home in the cabinet. So make sure the number on your microscope matches the spot that it's getting put away at. Whenever we are carrying microscopes, it's very important that we carry them by the base and the arm. Okay, I typically put one hand on the arm here, one hand cradling the base. Okay, and I rest it against my body as I walk it to the table. Once you walk it to the table, you can plug it in, and you are ready to get started focusing a specimen. So, there is kind of a procedure to focusing a specimen. You might think using a microscope is straightforward and easy, but there are some tips and tricks, and it can be kind of difficult to get things into focus. But if you follow these steps, it should be pretty straightforward getting your specimen into focus. So we always start with the scanning objective. Okay, this is the lowest power magnification because this is what's going to make it easiest to see whatever specimen is on your slide. So turn on your microscope, click on the light, okay, place your slide into the little clip on the stage, which I'll demonstrate in class. Place the scanning objective, click that into place. Then put your specimen in the center of your field of view. So that means get your specimen that's on your slide kind of to the center so it'll be easy to see. From there, you use the coarse knob, so that's the one that moves your stage quickly, and you're going to bring your specimen into focus until you get it perfectly crisp and clear. You can do this using a combination of the coarse and the fine focus knobs. So once you are focused, okay, then, only then are you ready to move up to the next magnification. Okay, don't touch your slide, don't touch your stage, don't move any of that. All you want to do is click over the turret, okay, and move to the next highest objective, so low power. Okay, you just repeat those steps. Okay, use the knobs to bring it into focus. Only once it's focused do you switch to a higher power. So you bring it into focus on the low power, don't touch anything, move to the next highest objective, and switch to the higher power. Okay, now what is key though? At the high power, okay, at the high power objectives, only use the fine focus knob. Okay, we're going to see this in class, but When you get to those higher power objectives, it's going to look like your objective lens is almost touching your slide. Now, it is not touching your slide, but the problem here is if you use the coarse focus, if you move that stage very quickly, you can crack your slide, okay? That you can shove that objective lens right up into the glass of your slide and crack it. So, recap. Only use the fine focus knob at the high power. Okay, low in scanning, you can use the coarse focus and the fine focus, but at the high power, we switch to fine focus only. Now, I've found that if you're at a higher objective, okay, low, high, any of those that aren't scanning, if you're out of focus, especially at the high power objectives, you're probably never going to be able to bring it into focus because once you're out of focus at a higher objective, it's really, 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 really tough to bring it back into focus. So what I do is I always move to the lower objective and start over. So if I'm on low power and I can't find it, I go back to scanning. I focus it there, and only once I'm focused do I move to the next. If I'm on low power, if I'm on high power and it's not focused, move to low power, focus it, then switch to high power and just make tiny little adjustments with the fine focus knob to bring it back into focus. So I've mentioned field of view, but what exactly am I talking about? So we call what we see when we look through a microscope the field of view. So it's going to look something like this over on the right. So it's a circle. Whenever you look through a microscope, you are going to see a circle. And you'll notice also we have a little pointer. Okay, not all of your microscopes will have this, but this makes it really helpful to point out specific things in a slide because you can angle it toward the end of the pointer and it'll help you show things to other people. But this is the field of view. Now you're going to see stuff in that field of view, say a little animal cell, a little nucleus. Okay, so pretend this is a little animal cell. You're going to see those cells within this field of view. Now, as we increase magnification, our field of view decreases. So what we mean by that, think about taking a photo with your phone, right? If you're fully zoomed out, you may be able to get a person's entire body in the photo, right? All the way feet to head. However, as you start to zoom in, right, as you pinch to zoom in on that person, you see less and less of that person, right? You might just zoom into just their torso, just their face. Okay, so we see less of them. Our field of view is decreasing as we are zooming in. Now, another kind of tricky thing that our lenses do is they invert the images that we see, which means whatever you are seeing in your field of view is upside down and backward. Now, it doesn't really matter too much until you're trying to move your slide around and find certain cells or for certain structures. Because if you're trying to move right, you gotta move your knob left. Yeah, it can get a little confusing, but that's what today and this lab is for. Okay, it's all about playing with microscopes. So we'll get used to that. The next thing we want to cover is talking about wet mount slides, because as part of our microscopy lab, you guys will be making your own slides. So one important part with this is how you place the cover slip. So the cover slip is this little piece of either glass or plastic. It's a little square, okay? And it basically holds our specimen in place. So whenever we're making a wet mount, you get your clean slide, you place a drop of water on the middle of that slide, put your specimen into that water, and set your cover slip on top of it. Okay, cover slip kind of holds everything in place, makes it nice and compressed. However, placing that cover slip, there is a trick to it. Okay, I ask, have you ever put on a phone screen protector? If you put it straight up and down, right, if you just slap it on there, what happens? What do you see? Very likely you see bubbles, right, underneath that phone screen protector because you've trapped air under it. And now you have a bunch of annoying bubbles and it makes your screen hard to see. So same thing can happen with a cover slip. If we just place it on flat, you're going to trap a lot of air. And because of that, it's going to make your slide hard to see. It's going to cover stuff. So if we place it down at a nice 45-degree angle, so you set it down and then you slowly we slowly let it drop at that 45-degree angle, just like putting on a phone screen protector. That is going to help reduce the amount of bubbles that you have on your slide, and it's going to make things a whole heck of a lot easier to see. We will also be staining our specimens. Now, so scientists use a whole bunch of different stains to see different parts of cells, right? It's like dye. Okay, we need to dye different parts of the cells in order to see them under a microscope. And we can use different stains to target different parts of cells, which is very interesting. But today we'll be using a stain that's going to dye anything that is negatively charged in our cells, in our molecules. So the dyes we are using, iodine and methylene blue, which we'll talk about more in class. But you might want to ask yourself and start thinking, what areas of my cells are going to get dyed if our dye targets negatively charged molecules? Okay, I'll let you guys chew on that one a while. So for this, it's similar to a wet mount. You're going to, instead of putting water, put a drop of stain on your slide, place your specimen, and then place your cover slip using our 45-degree angle procedure.